Hi, my name's Daniel Hubbard, and this is the research that I'm working on with Dr. Duffy. We synthesized a novel polycyclic phosphole, which glows blue under UV light. Um, Technically, this is a replication of what last year and the year before those students have been working on, but our reaction here is we're taking these conjugated rings and we're making another ring in there with a phosphole, and this stays aromatic. And polycyclic aromatic compounds are really good for producing fluorescent substances, and especially when you involve phosphorus in the mix. The homolumu gap, it it's enough. It allows you to fine tune uh, the emissiveness and the properties. I'm not sure exactly how it works. This is kind of complicated, and not a whole lot of research has been done on these, largely because of how difficult to make they are. So basically, the goal of this study was to um, produce a replication of what they had done before, and then build upon that and figure out stuff about this really interesting molecule. And because it's so unstable, we had to synthesize this underneath nitrogen gas. We couldn't have oxygen around until the very last step. And this is what we thought that we're making here, which is 7-phenyl 7H benzo E naphtho 2,1 beta phosphindole 7 oxide. So that's a mouthful, but this is what it looks like right here. As you can see, you've got your phosphole, and this was produced by taking your dibromobinaptylene, adding N-beta lithium to knock that off, put it back there, then phenyl dichlorophosphine, yeah, dichlorophenylphosphine, and then hydrogen peroxide, and that's what turns this into that. And after we synthesized it, we sort of did a little purification on it, but then we ran a column. And this right here, column chromatography, we ran it through with DCM and a certain percentage that we chose of acetone just to speed it up through the column. We chose 10% based on TLC analysis beforehand, and then we collected everything that went through the column, and we kept it in 60 separate test tubes, and then we ran TLC on each test tube. And of the 60 test tubes, 25 through 33 showed the most emissiveness. They glowed blue under low powered, and this brilliant cyan color under higher powered UV. So we believe that we made what we were setting out to make because of the emissivity. However, until we run an NMR and a mass spec, we won't know for sure what we made. And we made a couple mistakes, but it doesn't seem to have affected the product. We thought that oxygen might have gotten in once, and we kind of did a couple steps out of order, but the latter, that's not really a big deal. And since this is so new, and it's a new compound, right? Um, new new stuff. We gotta look into the properties, and like I said, we won't be sure until we know more about it, and that's what we're trying to do, is learn more about it. Future work, um, I was told that it'd be interesting if we shut this ring here and produce another phenyl group right there. I wonder what that would do. Also, um, even though we ran a column, I've been speaking with some people from the bio department to see if we can run a HPLC on it to get it really pure, and then that could help us find out more. But yeah, thank you to the chemistry department, Dr. Duffy, and my lab partner, Jeff. Thank you for watching my presentation, and I'll leave you with this really cool TLC we ran. It just glows. It's amazing. All right, thanks. Bye.